Dr. Andy Paterson is going to be talking to us about a brief history of plant genome evolution.
So some of my earliest childhood memories are of the fruit and vegetable gardens of my parents and grandparents. I just couldn't get enough of Pop-Pop's raspberries, of Grandma's applesauce, or of Mom's tomatoes. Moreover, even as a child, I was fascinated by reading seed catalogs, looking at pages and pages of different cultivars of tomato and cucumber and pepper in different sizes and shapes and colors and properties. This interest in plant genetics pervaded my education. As a graduate student, I learned how to breed crop cultivars. As a postdoc, I learned how to apply DNA tools to accelerate breeding of crop cultivars. And my lab has continued in these activities, developing and implementing tools for crop improvement in collaboration with breeders of the crops that provide us with our briefs, our currency, our vegetables and oils, our grains, our fibers, and other medicinal products that are essential to life. In the course of this work, we build up quite a lot of DNA information, eventually building up enough that we could begin to literally connect the dots between the genomes of different organisms, such as our major crops and botanical models, and even begin to make inferences about the nature of the common ancestor of all crops that somehow managed to evade being eaten by a dinosaur 150 million years ago. Through the course of this work, we found again and again the importance of a phenomenon called polyploidy, which is a genetic accident that results in a genome being multiplied twice or 3 times or 4 times or sometimes even 5 or 6 times or more, literally overnight. Polyploidy is very common in angiosperms, more common than in any other taxon known, even to the point of the crowning glory, which is canola or Brassica napus, that's multiplied its genome 72 times relative to the genome of the common ancestor of all angiosperms. Our ongoing work continues to focus on trying to fill in the gaps in this picture of angiosperm evolution, connecting more dots from more genomes, while also continuing to contribute to conventional crop improvement. A particular interest is in studying very recently formed polyploids, where we hope to be able to see literally in real time the processes and events that have given rise to botanical diversity, enabling the angiosperms to become the planet's dominant vegetation and also to provide essential ecosystem services. Thank you.